Hi everyone, I'm Carolina from Making Life, and today I'm here to interview Massimo Pedrani, a long-time member of AFI Scientific Committee.
Good afternoon, my name is Massimo Pedrani. I've been working for 42 years, unfortunately I'm getting very old now, in the pharmaceutical science. And I get my degree in chemistry and pharmaceutical technology. And spend most of my life in R&D development, galenical development, and above all to develop a new therapeutic system in the field of pharmaceutical science. What else? That is the aim of my job, and I perform a lot of, I would say, drug delivery system in this area. And I'm here to answer to your question, please.
Yeah. So let's start. Your session in Simposio AFI will talk about the formulation and the administration of medicines for the pulmonary tract diseases. So let's talk about the production of this kind of drugs that are composed of inhalation powders. What are the main challenges that you have to face from a technological point of view during their formulation, since you have to respect very strictly criteria like I think the dimension of the molecules? And why is it so important to have an interdisciplinary approach?
Yeah, that system is a very peculiar system, as well as most of the medical devices in the world require very strict, I would say, technical issues to be solved before to go to in the preclinical and clinical study. Let's say that basically the different techniques used to develop this kind of system included sort of technological approaches like milling, like freeze drying, spray drying, and supercritical fluid drying. So that means that you have to add many technology to do that, and there are not a unique technology to organize, to produce, to manufacture, and to develop this system. Why interdisciplinary? That's very important, not only for pulmonary delivery, of course, because the new approach in the delivery system requires to have different expertise in the field, the galenical, the manufacturing, but not only the analytical is very important for the quality of a product and production for the quality of a product. And that's not sufficient to have a good system able to go to the market, because they require, of course, some preclinical study, toxicological and safety, which is the second part or the first part, whatever it is most important to have it. And the third one is the clinical, of course, to able to introduce the product through the different phase clinical study to well recognize, to demonstrate the efficacy of a system. That's the reason why interdisciplinary is fundamental for this kind of system for any kind of drug product, in particular for pulmonary delivery system, because it's very complicated, very complex approach.
I understand, and I think it's very interesting, the preclinical phase. So what are the difficulties in the switching between the in vitro studies, so on cell lines, from the in vivo studies on animals? The difficulties are because of the typology of the administration?
That's a very good question. You know, that's the difficulty for any scientist in pharmaceutical sciences to have a model to try to transfer what you do in vitro and what you do in vivo. That's a very big question that we have for many drugs, for many products, not only in pulmonary, of course. So we have to include also the administration route. In this case, is very peculiar, because to inhale the product is very difficult to try to have an animal model. But anyway, probably is much better to use some cellular model just to demonstrate the adhesion of a system in the epithelium. That's another target to be considered very useful to demonstrate the quality of your product. So your question is just how to complain or to explain how to demonstrate the in vitro in vivo correlation. And the second issues that complicate this study is that whatever you do in the healthy volunteer is completely different when you compare with patients, because the inhalation system is affected from the disease. So that's the reason why when we have something in vitro that's working very well, and we use transfer in a pilot study or in a phase 1 study in humans, in human body, there is a big difference between healthy volunteer and the patients. So it's quite complicated because the respiratory tract is affected from the inspiration powder in particular. You can imagine how it's complicated to have a good model in this case. So that's the reason why you are quite obliged to use the biopharmaceutical just preliminary for quality issues, not for efficacy issue, because that's required full clinical development, included the patient disease in different disease.
Yes, so this is why the concept of bioequivalence is not applicable in this case.
There are some guidelines. EMA has a different guideline if you compare with Food and Drug Administration, but there are some guidelines that can be applied to try to have some bioequivalence study. You can imagine that the bioequivalence basically is something that is related to plasma level for the pharmacokinetic study. And sometimes it's not easy to have some significant plasma level through this kind of administration route. However, you can imagine as part of a drug is absorbing the upper part of the respiratory tract. So you have something is similar to the oral release, and so you can see some plasma level to try to demonstrate the absorption of the drug. Unfortunately, something it is not completely easy to do, but you have to follow guidelines. It depends, of course, on the type of device. It depends on the type of disease. One thing is working with asthma, another thing is working Alzheimer or whatever it is, which is another disease that this device could be applied. So you can imagine it's very complicated. There are not a single specific way to approach the product. You have to adapt to the kind of disease, to the kind of administration, to the kind of drug substance, because one thing is to work with budesonide, another thing is work with other derivative of budesonide or other product that could be administered by this inhalation route.
Yes, I think it's very difficult just to correlate the models, because a person inhalates volunteer, a rat, a mouse have a different
model,
and I can think about the in vitro model, because you are administration the substances to the cells, so they are not inhalating anything. So it's very difficult to translate this kind of drug experimentation.
Right. That's the point. But anyway, basically, for all the company that like to introduce this kind of device in the world, are quite obliged to apply to the patients, so related to the disease. Again, full clinical trial is quite recommended to do to demonstrate the efficacy of the study. Of course, you require to have multidisciplinary activities as well, as we said before, because that's something that allows you to introduce in the clinical trial medical device.
So my last question is about a hot topic that we are talking about right now, so the sustainability. The production of this kind of drugs with this kind of powders requires a particular attention to the environmental impact and the environmental protection.
Well, that's a very good question. Let me share the question in two different answers, because one thing is to work with a new drug substance in this kind of device. That's oblige you to follow the official guideline for environmental assessment. Okay. That's the one thing, because it's a completely new drug. If you use a well-known drug administered with the new device, you have to consider that one thing is to work with the pressurized metered dose inhaler that requires hydrofluorocarbon, HFC derivative that can really affect the environmental disease. Okay. The powder the device is different because the impact on the environmental is lower. Is very lower because you can imagine we have powder versus the fluoro derivative that's impact with the atmosphere, impact with the environmental approach. And so I repeat, one thing is a new drug, one thing is a new system with the excipient of a propellant that you have to use to apply to specific drugs. That's the different approach. That's the different impact in environmental aspect.
Thank you very much. You were very, very clear in your answers.
Thank you.
And I hope to see you at the AFI Symposium.
Thank you very much. You are very welcome. See you soon. Thank you.
Thank you. Goodbye.